Welcome back. This is part 2 of the brain, which is actually part 1 of chapter 12. So that's a confusing way to do it. I apologize. This will wrap up the brain information though. So let's do it. So we're starting off here with cerebral white matter. It's kind of a good spot to put part 2 because before this point we were talking about gray matter. We were talking about regions of the cortex and what they do, what their function is. Now we're talking about white matter. White matter is composed of axons. Axons are the tail of the neuron. So they are concerned with action potential propagation. You can think of them as copper wires. The analog to the gray matter would probably be like the hard drive or the computer chip or the processor in a computer. And this white matter is like the electrical wires that connect all those components. So we can name that wiring based on what direction it's headed. So we have 3 types. Let's take a look. Number 1, this group of axons are called association fibers. These are axons that run from one gyrus to another within the same hemisphere. And the name makes sense, association, because a neuron, an axon that connects the primary somatosensory cortex to the somatosensory association cortex, that's going to be called an association fiber because that's one gyrus to another within the same hemisphere. Now, number 2, these types of axons are called commissural fibers. So these are axons that carry information from one side of the brain to the other. So if it carried information from the right somatosensory association area to the left somatosensory association area, that would be a commissural fiber. Now, if you remember the corpus callosum from lab, this was made up of commissural fibers. And in this photo here, the corpus callosum is sort of outlined in red. And I forgot to mention this picture, I'm sorry. This is something, I believe it's called tractography. It's done with an MRI, and they can map white matter. And then it's color-coded based on transmission direction. So that kind of violet-purple color you see sort of hidden behind the blue and green, those look like they're probably association fibers. The red are commissural fibers. And then the deeper blue and the green look like they may be projection fibers, which is our last group of fibers. These are fibers that carry information from one level to another. So from the cerebral cortex to the cerebellum, or from the cerebral cortex to the spinal cord. They move up and down the central nervous system. All of this is considered white matter because these are axons. White matter is composed of axons. Our next group here is called the basal nuclei. You've probably heard the term basal ganglia. This is actually gray matter. Basal ganglia is not technically an accurate anatomical word because a ganglion is a group of cell bodies outside of the central nervous system. And you can see from this picture that this gray matter is within the brain. So it's certainly within the central nervous system. So the correct way of describing this tissue would be as a nuclei. So basal nuclei is technically the correct term. It just takes time for these things to change over, so you still might hear basal ganglia. There's multiple nuclei within the brain: caudate nucleus, putamen, globus pallidus. These are all basal nuclei, and it's gray matter, just like your cerebral cortex. Now this is straight out of your book. Their role is misunderstood, but thought to act in filtering impulses related to movement, cognition, and emotion. And I mean, I don't know that I would say that their role is misunderstood, but I mean, to be fair, when it comes to the brain, yes. Often we are extrapolating, we're looking at data and we're drawing conclusions from that data, and sometimes it is flawed. So I think it's probably fair to say that a lot of the brain is misunderstood. But yes, it is true that this part of the brain acts to filter impulses. It acts to filter impulses. And one thing that makes that clear is with certain basal nuclei disorders, you may have excessive movement or impaired movement. So it does kind of give credence to that idea that these fine-tune and filter impulses. And not just movement, although that's the most common, but also cognition and emotion. Really quick, if you guys want to see something really cool, I should have looked this up before I started, but look up a pallidotomy procedure at Loma Linda University, or at the medical center on YouTube, and everyone has perms and big mustaches. So I'm imagining it's from like the 70s or 80s, I don't know. And they show a guy come in with Parkinson's, and he is essentially cured. He can't walk. He can't touch one finger to another. He can't walk. He can't move through a doorway. It's a common Parkinson's sign. Even if you can walk, you can't walk through a doorway or over a change in texture on the floor. If you switch from carpet to wood, that does something, and it's difficult depending on the patient and the disease process. But anyway, so he has a pallidotomy, and he comes out of the operating table hours later and jumps up. He's running around. They show a follow-up video. He's like doing cartwheels. It's crazy. So it's great to look at. I always mean to do a deeper dive into why they don't do this procedure anymore. I assume there are some significant risks or side effects or who knows. I mean, that has to be the case. Otherwise, I don't imagine why they would have stopped doing it. But check it out. See if you can find the video. I'll try to post a link on Canvas. Okay, so we've talked a bit about the diencephalon in lab, but the diencephalon is made up of the thalamus, hypothalamus, and epithalamus. The thalamus, we're going to actually look at the thalamus somewhat in depth next week. The easiest way to describe the thalamus, it's a bit superficial, but I would call it a sensory relay center. Now, every sense that comes, well, just every sense period with the exception of smell, I suppose, goes through the thalamus. And there's filtering that occurs there. There's a rerouting. There's integration. That's the best word. There's integration that occurs in the thalamus. And then that impulse is wired to the appropriate part of the brain. So that's where all incoming sensory information into your brain is handed off and then delivered to the right pathway to the right part of the cortex. So you could say that it filters sensory information, it routes sensory information to the appropriate location. Then you have the hypothalamus. The hypothalamus really regulates the pituitary gland and other aspects of endocrine and your system's physiology, endocrine function and physiology. It tells the pituitary gland what to do through tropic signaling, which just means hormone signaling. And it also actually has a direct nervous system connection to the posterior pituitary gland. So it's very involved in autonomic function and endocrine function. A very kind of short note to jot down is that it maintains homeostasis through various mechanisms. Autonomic reflexes are involved here. You know, if osmoreceptors in your aorta sense that your blood pressure is low, the hypothalamus may tell the pituitary gland to release antidiuretic hormone, which causes you to retain water. So that type of idea. But for the purposes of this chapter, you can just write that it maintains homeostasis. And then you have the epithalamus, which is technically a gland. It's called the pineal gland. And what's pretty cool about the pineal gland is there's only one sense of your special senses that connects, that's kind of the wrong word, but that has pathways going to the epithalamus to regulate its function. So it might help you if I tell you what the epithalamus releases, and it releases melatonin. So then if you put your thinking cap on, what special sense should regulate melatonin release? Well, not smell, of course, because what would smell? You smell something smelly and you get tired? That doesn't make any sense. Would it be taste? Would it be hearing? You could maybe make an argument for hearing, I guess. Like, hey, it gets quiet, so it's time to sleep. But what's something that happens every night before we start to get sleepy, unless you're an insomniac like me? The sun goes down and it gets dark. So you have visual pathways, neurons from your retina that through a chain, through relay, make their way to the epithalamus. And daylight inhibits the epithalamus. So when your eyes are activated by blue light specifically, so that could be from the sun or from an LED light bulb or a fluorescent light bulb, melatonin release is inhibited. Phone screen, that's where the whole blue light filters come from on phones, is they realize that blue light inhibits melatonin release. So if you're on Instagram all night in bed, your body's not releasing any melatonin. Maybe you've taken 10 grams of melatonin, so you still fall asleep. but anyway, so and people at this time, people often ask in class, what do you think about melatonin? So melatonin in your brain definitely makes you sleepy. The question is, how bioavailable is supplemental melatonin? And what that means is, how readily absorbed is it? Is it converted to something else by stomach acids? Is it absorbed in its active form? And then if it is absorbed, does it actually make its way across the blood-brain barrier and into your brain? I don't know. I should look up studies on that. I apologize. I owe that to you guys, and I haven't done it. But that would be, if you were curious yourself, that would be where your research would go. You'd go to Google Scholar or something, and you type in melatonin and sleep, and you'd read. And so they'd probably do one of two studies. They'd actually test to see what happens to your melatonin concentrations in your blood after supplementation, and then they would probably do a test where they maybe do sleep studies on people and look at their EEG, or they just have a survey and they ask people to rate how many hours they got, blah, blah, blah. So there's lots of different ways you could look at that. So check it out and get back to me. I'm curious. I have done some melatonin literature review on human growth hormone release, though, and melatonin is linked to endogenous human growth hormone release, but so is sleep. So the question is melatonin causing human growth hormone secretion, or are you getting better sleep and therefore getting better human growth hormone release? And that's actually a part of my research study I'm designing. So don't do a study on that. Don't beat me to the punch. Okay, sorry, I'm rambling. Let's go to the brainstem. So the brainstem sits right below the diencephalon. It's composed of the midbrain, pons, and medulla oblongata. The midbrain has various reflex tracts, and there are some nuclei in the midbrain. We're not going to go into a lot of depth here. The pons has a lot of conduction tracts. There are also a few cranial nerve nuclei within the pons, and then we have the medulla oblongata, which is an autonomic reflex center. So if you stand up and your aorta senses a decrease in blood pressure because you just stood up, right? So your blood is sinking, so to speak. It's not that your blood is sinking, but that your pressure drops in the upper half of your body when you stand up. So in response, your heart starts to pound harder, more forcefully and more rapidly to get blood to your brain. And then depending on how low your blood pressure is, you might get a little lightheaded when you stand up, and depending on how relaxed you were. So the medulla oblongata is an autonomic reflex center. Pretty similar to the spinal cord, but not identical, and also its function, the brainstem's function, is much different. Any damage to the medulla oblongata is a big problem. Any damage to any part of your brainstem is a big problem, but the medulla in particular. A lot of times that is actually what causes death in a traumatic brain injury, is you get cerebral edema following the accident, and as pressure builds within the cranium, the brain stem is pushed inferiorly and eventually will make contact with the anterior rim of the foramen magnum. So the medulla oblongata can actually make contact with the foramen magnum, and neurons do not respond well to compression. They're great at tension. If you've ever stretched and touched your toes and felt a little tingling in your toes or behind your knee, those are your neurons being stretched. But that doesn't feel great, and you can certainly overdo it, but it doesn't destroy neurons. Compression of cell bodies does. So neurons do not like compression. That's why hemorrhagic strokes are so dangerous. Oh, I'm sorry, guys. I did not realize I had a separate slide for each of these brainstem regions, so I wouldn't have gotten so much detail in the last slide. So cerebral peduncles. Can't really see them too fantastically because this is a posterior view. We can see some cerebellar peduncles Yeah, we need to have an anterior view to see the cerebral peduncles, but the peduncles, any peduncle, is a big bundle of axons that turns into a spinal tract if it makes its way into the spinal cord, and it's like a big transmission line. So the cerebral peduncles are a part of the corticospinal tract, which anytime you move any muscle in your limbs, you're utilizing that tract. So I won't ask about that on the quiz, I don't think, so don't worry too much about that. Then we have our periaqueductal gray. This is a collection of gray matter around the central canal of the brainstem, and it doesn't extend to the spinal cord, I'm sorry. So it's just in the brainstem. This is actually the part of your brain that Tylenol, acetaminophen, acts on, I believe. I hope I'm correct about that. I think I am. It acts on the periaqueductal gray matter to manage how you perceive pain. Then you have the superior and inferior colliculi. These are reflex centers. Particularly, this is where the integration occurs of the reflex. So the superior colliculi are visual reflex centers. If you see something fly, your eyes have a tendency to follow it. That's a visual reflex. If you hear a loud noise, you have a tendency to look at it subconsciously. Like a lot of times you hear a loud noise, you're looking at it before you're really even aware of what's going on. And a lot of times you may close your eyes as well in response. So that's the inferior colliculi that do that. Together, all four of those nuclei are called the corpora quadragemma. Again, I probably won't ask you about that. Substantia nigra, this is a gray area within the midbrain, I believe. It's involved in Parkinson's disorder. You have a dopamine deficiency that develops there for whatever reason, and you get Parkinson's syndromes. The red nucleus is it's a part of a motor pathway. I don't recall exactly which motor pathway. I know it has to do with posture, and I think it innervates flexor muscles of your trunk. I don't even remember, so of course I won't expect you to. So let's keep going. The pons is composed primarily of conduction tracts. You have vertical or longitudinal tracts, and then you have transverse tracts. So the transverse tracts go back to the cerebellum. The cerebellar peduncles you saw on the last slide are a part of this, and then you have projection fibers that run from higher brain centers down to the spinal cord through the pons. Those are running longitudinally. The medulla contains what's called the pyramids, and there's also the olives, which are a part of an olivary nucleus. We won't cover that, but the pyramids are a continuation of those cerebral peduncles, which you can actually see in this photo right here. You see these kind of stranded, flat cables there. That's axons coming from the primary motor cortex, and they continue down to the pyramids. That's why it's sometimes called the pyramidal tract. Anyway, at this level, the pyramids cross, and that crossing is called a decussation. So we say that the decussation of the pyramids is located in the medulla oblongata. And if you are a Waterboy fan, you also know that this is why alligators are so angry. It's not because they don't have a toothbrush. It's because of their medulla oblongata. And if you've never seen that movie, you have no idea what I'm talking about, and I apologize. It is an autonomic reflex center. Cardiovascular reflex centers are there. We discussed that. Also respiratory centers. So if your blood is becoming a little acidic, you may increase your respiratory rate. That reflex would occur here. It would occur in the medulla oblongata, and there are other autonomic centers as well. Then we have the cerebellum. 11% of brain mass. So between the cerebellum and the cerebral hemispheres, you're at 94% of the brain. We've traditionally thought of the cerebellum as a coordination and reflex center. So you guys don't probably, maybe you do, I don't know, but you probably don't realize how much of your brain is devoted to movement. We would expect like one lobe would be for math and one lobe would be for language and one lobe would be for art, but that's really all constrained to your frontal cortex. Of course, you know, you have some sensory areas in your temporal and parietal lobes as well. But so much of your brain is actually dedicated, and I'm sorry I said frontal, I meant prefrontal. So much of your brain is dedicated to movement, and we used to think that was all the cerebellum did. Just imagine if you're a hurdler, when you're in the Olympics, you're sprinting, and then you have to hurdle over those tall things and land, and there is just an incredible amount of data being picked up by the receptors in our body. So your muscles are packed with what's called muscle spindles that measures length and rate of length change. Tons of data coming from every muscle. You have Golgi tendon organs monitoring stretch in your tendons. You have free nerve endings all over the place in your skin, around joint capsules. You have Merkel disc, Basinian corpuscles. You have any number of receptors all throughout your body. And as you're running and you're jumping, you're getting information sent to your brain about how long each muscle is, how much stretch is on it, how much tension is on the tendons, how quickly the change is happening. Is it elongating really fast or is it elongating relatively slowly? That all gets fed into your brain immediately. It's so fast. And then your brain, your premotor cortex, and your cerebellum make plans on how to adjust and adapt and increase tension of this muscle and increase tension of this muscle really quickly. Increase tension in this muscle, but allow it to elongate as you land while you're increasing tension. It's really, it's nuts when you think about it. You think that math would be the more difficult thing that our brain can do, but it really is coordinated and complicated movement. It's pretty amazing. So we just kind of assume that's what the cerebellum did, and it does. But we now know that it doesn't just coordinate physical things. It also coordinates any complex task. So any complex emotional environment or decision or setting, our cerebellum gets involved. Any complex cognitive issue, our cerebellum gets involved. So it's more like an auxiliary processor, although it does tend to be utilized for movement primarily. So anyway, real quick, I won't ask you about this, but just a very simple layout of cerebellar processing. So remember, movement is initiated in the cerebral cortex, possibly the primary motor cortex if it's a really simple movement, but more likely the premotor cortex. At the same time, the cerebellum is actively receiving and monitoring proprioceptive input from throughout the body. If you don't know what proprioception is, I thought that was a common word, but I have a lot of students who had never heard it. It's just the idea that if you close your eyes and I move your arm, I extend it straight out from your body, you know where your arm is, even with your eyes closed. If I put it behind your back, you know where your arm is. So the point is you can feel the position of your joints, your muscles, your tendons, your ligaments, and that information is being sent to your cerebellum. So the cerebellar cortex, the premotor cortex most likely, calculates the best way to coordinate the force, direction, and extent of contraction to ensure smooth movement based on the information the cerebellum is monitoring and relaying to the motor cortex. This blueprint that's developed in coordination with the cerebellar cortex and the cerebral cortex is delivered to the primary motor cortex to be delivered to the muscles. So pretty complicated process. Okay, there are functional systems within your brain. We're going to talk about two of them. The first is the limbic system. This is oftentimes called your affective or your emotional brain. So this is a part of your brain, if we were to put you in an fMRI and make you sad or make you angry, this is a part of the brain that would light up. It gets involved in emotional situations. But what's so interesting about human beings is that most things are emotional for us. We apply emotion to cognition more than an animal would. Even a very logical task, emotion tends to get involved. So the limbic system is made up of all sorts of different components. the cingulate gyrus, the amygdaloid bodies, the hippocampal formation, the fornix. There's more, but a bunch of different structures are involved. I don't remember exactly. Oh, we have a slide for the limbic system. Okay. I was like, I don't remember all what I was going to say about the limbic system. So it's our emotional brain. The amygdaloid body responds to perceived threats with fear or aggression. Man, you have to monitor that amygdala. If you feel yourself just getting annoyed with everything and getting angry and defensive, you've got to do some amygdala therapy. You've got to calm down. It's not good for you. We'll talk about that in the endocrine system. So work on that. It's not good to be angry or afraid all the time. I mean, nowadays that's tough. It's very tough. Sometimes you got to get angry though, but you don't want to live like that all the time. Why am I preaching to you guys? I'm sorry. The cingulate allows us to express our emotions with gestures. Isn't that interesting that we have a specific part of our limbic system that allows us to add emotional context to our words with gestures? That's pretty cool. And I meant to mention this earlier, but so, a lot of times when I'm talking about an area of your brain like Broca's area, that's just on one side of your brain. If you're right-handed, it's on your left side of your brain. So then what's in that exact same spot on the other side of your brain? Because anatomically they're identical. If you just look at the surface of the cortex, it's going to be the same on both sides. So how can you have this area that produces speech on one side, well, what is on the other side? And what's so interesting is that it's often related. So in the exact same spot where Broca's area would be on the right side of your brain is a part of your brain that helps you give tone to your voice, and or maybe even goes beyond that. Maybe it helps you gesture, like the cingulate gyrus. It's involved in body language or facial expressions. It's a link to language production. I just think that's interesting. Anyway, our sense of smell is highly linked to the limbic system. If we go back and look at this last picture, these two things right here, these are your olfactory bulbs, and then that's cranial nerve 1. They feed in directly into your limbic system. It doesn't go through the thalamus like your other senses, which is pretty interesting. And that's why they say, smell can make you emotional. And I've always not really agreed with that. But I remember I walked into a classroom from high school. I hadn't been there in, oh boy, 15, 20 years, I don't remember. And I walked in there with my wife, actually, who I met in high school, if you can believe that. And I just had this like wave of memory come back when I walked in and smelled the classroom. I remembered the smell. I couldn't have told you what that room smelled like beforehand. I had no idea. But then when I walked in, I was like, oh yeah. And it wasn't like it was a bad smell or a strong smell. You just smelled a certain way. So anyway, it was linked to memory, which is a part of your limbic system, which makes sense. That memory and emotion would go hand in hand. But it also makes sense too. We react very strongly to smells. If we see something really blue, we might go, "Oh wow, it's beautiful." But if we smell something that smells really terrible, we're like, "Oh my goodness," we're upset. What is that? Oh, it's the worst. Or if you smell something amazing, baking cookies, you're like, "Oh my goodness, that smells so good." So it's a minor thing, but I think you probably agree that smell, for whatever reason, can be it's more of an emotional sense. The hypothalamus is right next to this and is actually somewhat integrated with the limbic system. And so that is one hypothesis behind some psychosomatic illness. If you're just emotionally troubled over something, or something is just very traumatic, it can actually make you sick because there's just some crosstalk. There's so much action potential and ion flux that you can get that psychosomatic illness. Anyway, the cognitive and emotional brains are highly integrated. That's an understatement. Even a lot of us who kind of like to think that we're cold, logical computers, it's tough to separate that part of us for sure. So, and maybe we shouldn't try. So I think I'm feeling like profound today. I don't know why I'm talking like this. I apologize. The reticular formation and the reticular activating system. This is a more difficult system to visualize. Here's a sort of diagram of some of the pathways, but it's not a clear-cut anatomical structure like the limbic system is. So they're just showing that the eye has something to do with it. Pain, temperature, and touch receptors from the body have something to do with it. Your input from your ears have something to do with it. That's all true. But what is it? So it's kind of a scattered network of neurons throughout the brain. And these axonal connections come from all over the body, and they connect all over the brain. A specific subset of these is called the reticular activating system, and they are constantly sending a stream of impulses to the cortex. All ascending neurons actually synapse on these neurons. A branch of the axon synapses on a reticular activating system neuron, which stimulates them. So this is interesting. So you have these neurons in your brain that are constantly sending signals to your brain at a steady rate. But incoming sensory information stimulates it. So this is the idea that sensory stimuli, this is not like the accurate word, but wakes your brain up. It gets your brain going. And we've all experienced this if we've ever gone on a road trip or had to drive at night for a long amount of time, which I love. I didn't realize how few people enjoy driving. It's probably good, I guess, for the planet to not enjoy driving, but I love to drive. I drive a lot for work, so not right now, obviously, but if you were taking this class from me in person, we would meet Monday night, and then on Tuesday morning, I would hit the road. I'd go up to Oregon or San Francisco or, Sacramento, wherever. San Diego, that's close. I can do that in one day. But so Tuesday I might leave and I might come back on Friday, and then maybe my TA would teach lab on those weeks. So I enjoy that. I like it. I actually drove to Florida with my wife and my 2-year-old, if you can believe that. And everybody thought we were crazy, but we liked it. It was fun. It's just kind of nice. Anyway, but there's an element of risk with that. I mean, other than some people just drive crazy. There is an element of risk that you might fall asleep, because what happens is there's very little stimuli when you're in a car. And it's consistent stimuli. It's like the hum of the engine, the vibration of the road. It's consistent. It's just that little zzz. And your reticular activating system filters out weak or repetitive stimuli. That's the last bullet point here. So you start getting sleepy. And logically, you're like, my goodness, if I fall asleep, I'm going to die, right? So you think logically you'd be able to just convince yourself like, hey, I need to stay awake. But you can't. It's just a force that you can't fight. And if you've ever driven far enough or long enough or been tired enough, you've probably come close to falling asleep. Some of you maybe have even fallen asleep. I've come pretty close, and I hate to say it because I know it's so dangerous, but just being by myself on a longer drive, no other cars on the road, it's midnight, and every once in a while you get a bright light the other direction. It's just your eyes are kind of hurting, and then you might sort of start to drift to the right. And then if you've ever been on one of those big freeways, they have grooves on the right part of the slow lane. So if you hit them, it makes your heart go brrr, and that wakes you up, and you snap back to reality. And you probably even at that point you might say, okay, I need to listen to my music really loud. So you get loud music, and that wakes you back up. This is the system that does that. But then you listen to loud music for long enough, and it starts to become a repetitive stimuli, and you start falling asleep again. So you pull over and you get a cold iced tea, because that's a lot of sensory input. Cold iced tea, that's a lot of temperature receptors, taste receptors, so that wakes you up again. And you do that battle. I also think it explains why some people like to study at coffee shops. So I wish I could ask for a show of hands, but who likes to study at a coffee shop or Panera or something? And that is There is a divisive line. You either do or you don't. And so I love it because for me, being in that environment of stuff happening, plates clinking, people walking by, doors opening, having something hot to sip on, that stimulates the reticular activating system, and I can focus and I can study. Some people, that's distracting. Maybe it stimulates that system too much. So everybody's different, but anyway, so that's the reticular activating system. Its job is to stimulate the cortex. If its stimuli decreases, you start to become kind of lethargic and tired, and eventually you'll fall asleep. So when this system turns off, that's when you go to bed. You fall asleep, and then in the morning it fires back up again. It's more complicated than that, but we'll talk about sleep. Oh, we're talking about sleep in a few slides. My goodness, we're talking about it right now. So higher mental functions of your cortex, not just your cortex, but mostly of your cortex: consciousness, sleep, language, and memory. Your book's pretty funny. It says, how do you define consciousness? We know how the brain works. Ions are flowing in, they're flowing out, signals are being transmitted. But if you ever watch a sunset, you can't explain that with ions. And I'm like, yeah, I guess. It's just such a funny thing for a textbook to say. I agree wholeheartedly, though. It is very tough to explain what consciousness is. And here's your book's best crack at it. It involves simultaneous activity of large areas of the brain, of the cortex. Okay, cool. It's superimposed on other types of neural activity and is interconnected. This is the best non-answer. So what is consciousness? Well, it's a bunch of activity that's connected to other types of activity. So I like that. The point is, scientifically, it's tough to explain what consciousness is. But there's clearly a difference between being awake and being asleep. And that difference is consciousness, whatever that is, whatever that special something is, we have it. Thank goodness. Sleep is partial unconsciousness. So complete unconsciousness, that would be death. So I guess I shouldn't say the difference between being awake and being asleep is consciousness, but that's basically what it is. It's partial unconsciousness. Your cortex is suppressed, but your brainstem remains active. And this is so interesting. Environmental monitoring remains active. And that is why people are able to sleepwalk so efficiently. I've had some crazy sleepwalking stories from students over the years. But get this. This is what happens when you fall asleep. Oh, okay, first question. Do you know what the most common dream in the world is? Most people say that it's a sense of falling. I think that's probably true. I just don't know if that's considered a dream or not, I guess. But what I read, and you can't believe everything you read, but I think it was from somewhere trustworthy, some kind of study or something, was that the most common dream in the world is losing your teeth. And it's weird because I've never had that dream, ever. And I just had it a couple weeks ago. I had a dream that my teeth fell out. Oh my gosh. So weird that first of all, I dreamed about that, but number two, that so many people do. And that just must be a survival thing, just deep in our DNA. We worry about losing our teeth. I don't know. Anyway, where was I going with that? Oh, so actually, I think I might want to talk about this on the next slide. I don't want to jump the gun. Okay, let's hold that thought. So, what types of sleep are there? Well, there's two types: non-rapid eye movement sleep and rapid eye movement sleep. And that's pretty self-explanatory. During non-rapid eye movement sleep, your eyes do not move. During rapid eye movement sleep, your eyes are moving all over the place, which fortunately I've never seen. If my wife goes to sleep before me and I walked in there and I saw her eyeballs zipping around under her eyelids, I would be a little creeped out. I don't know, it just seems so weird, but we all do it. So anyway, so I'm going to talk about those a bit, but I actually think in a couple slides. So we have a sleep pattern. We have this alternating 24-hour sleep and wake cycle. I actually think it's a 25-hour circadian rhythm, but anyway, it's dictated by the hypothalamus. There's this nucleus called the suprachiasmatic nucleus, so it's right above your optic chiasm. That's why it's called suprachiasmatic, and it inhibits your reticular activating system. And a part of that has to do, if you're just relaxed enough, it will inhibit your reticular activating system. But the pineal gland plays a role as well. When no light is coming into your eyes, your pineal gland, your epithalamus, starts to release melatonin, and that stimulates the suprachiasmatic nucleus, which inhibits the reticular activating system. Anyway, within that rhythm, you have a REM cycle every 90 minutes, a rapid eye movement cycle. Your first REM cycle is very short, but they get progressively longer throughout the day. So here we go. We're awake. We lay down, boom, we fall asleep. So we're in stage 1 non-REM. We fall into a deeper sleep. Our brain waves start to change. We're in a stage 2 non-REM, a stage 3 non-REM, a stage 4 non-REM. At this point, we are knocked out. We are asleep. This is when like an earthquake could happen and you wouldn't know about it because you are dead to the world. You're passed out. This is when your mouth is open and you're just like you're unconscious. You're not dreaming during this stage. Interestingly, you're not probably thinking anything. Your brain waves the amplitude is big and the frequency is huge. These are just big, slowly oscillating brain waves. The metabolism of the brain is very low, very little oxygen consumption. And then boom, all of a sudden you rocket out of it. So between like at 90 minutes ish to before we get to 2 hours, we blast out of that and we go into what's called a REM state, a rapid eye movement state. Your brain is actually more metabolically active in REM than it is when you're awake, which is interesting, right? And these are high frequency, low amplitude brainwaves. Your brain is doing stuff during this cycle. I wish we could kind of come in as like a fly on the wall during somebody's REM cycle and see what's going on in there. Then you go back down to 4 and then back up to REM. And they don't do a good job showing this, but each REM cycle gets progressively longer. The one right before you wake up is the longest. And you might wonder, why do I always remember the dreams from the morning? And that's because you have this big, long, beautiful REM sleep right before you wake up. Sometimes your brain is so metabolically active and your dreams are so clear that you actually think you're awake. You can think to yourself, oh, I'm awake. And then you're like, I got to focus on this dream I'm having because my alarm's going to go off soon. And you can feel the covers. You feel like you're just awake, but you're actually asleep. You're in REM. And that's a fine line. You might actually wake up. But so that brings me to my next point, which is that non-REM and REM do different things. So non-REM sleep is restorative. Let me explain that a little bit. So I think a great way to explain it is by what happens when you're deprived of one type of sleep or the other. So if any of you guys have ever taken NyQuil or ZQuil or just diphenhydramine in general, Benadryl, which I didn't realize till like I started teaching, oh man, I think it was one of my pharmacology classes that Benadryl and NyQuil have the same active ingredients. Interesting. But so anyway, if you've ever taken that, you wake up feeling like, man, I was out. Like I just don't even remember sleeping. I was knocked out. You close your eyes, you wake up, you're groggy. You got probably a lot more non-REM sleep. So you got sleep. Like you're awake. You got a good night's sleep. But you're kind of groggy. You're kind of cranky. You might be a little short with people. You don't feel great, even though you got a lot of sleep. That's because non-REM sleep, that's what you need to just like physically function. To cognitively function and to physically function, you need non-REM sleep. To function emotionally, to have emotional stability, you need REM sleep. because REM sleep is emotionally restorative. And we don't really know exactly why it is. Your book kind of speculates that it may be an opportunity to process a day's events and work through emotional problems via dream imagery. Because REM is when you dream. You dream during REM. You don't dream during non-REM. And oftentimes that's why you feel like you only remember your dreams from the morning, because your first REM cycle's 20 minutes long. So you probably didn't do a ton of dreaming. But your last REM cycle might be an hour long or 2 hours long, depending on how your alarm clock timing plays into that, and that so you feel like you remembered that dream. So some things that might inhibit REM sleep are a lot of the kind of anti-anxiety drugs, all the diazepines and stuff, the benzos, Valium, and if you've known someone who is, I would argue, addicted to Valium, although it was prescribed in this case, they struck me as being sleepy but happy. So kind of just like, hey, how's it going? Yeah, I'm doing good. Yeah, eyes are half closed, but yeah, just like very like not reactive, but also lethargic. And I don't know if that's just a side effect of the medication, but it may also be because they're not getting any non-REM sleep. So it's difficult for them to function at a high capacity physically and cognitively, but they're not angry, right? So if you've ever had to wake up 2 hours earlier than normal, like to go to the airport or something, man, how cranky are you? You get used to whatever your sleep schedule is, even if it's shorter than it should be or longer than it should be. You get used to that routine, and then if it has to change all of a sudden one day, it affects you. If you have to wake up an hour early, you just missed out on an hour of REM, which is a lot considering you only get a couple hours of REM throughout the whole night. So you might be a little cranky if that happens. A secondary hypothesis on what REM sleep is reverse learning. I don't really know what that means, and I don't think your book did a great job explaining it, but I guess the idea is throughout the day things happen and stimuli occur and they really aren't relevant or helpful, so your brain has to get rid of them and undo them and not store them, basically. So anyway, I guess a good time to talk about sleepwalking never came up, so I want to circle back to that. What's so interesting about sleep and REM sleep is this. So why do your eyes move? And so basically what happens is when you fall asleep, your body produces and releases a paralytic. Thank goodness it's a temporary paralytic, and that goes into your blood and it makes you so you can't move that much. And if you've ever, this is a dream I always have, is I feel like I can't move in my dreams. Like I'm trying to run or walk and I feel sluggish. So I get a lot of feedback from students that happens to them as well. And this is probably why, because I can't. When you're dreaming in your sleep, you really can't move. I mean, you can. It's more difficult to move. And this is just to keep you from acting out your dreams. You don't want to be sword fighting and leaping and flying around. Uh, I just gave some clues into the kinds of dreams I had. I'm embarrassed. You don't want to be doing that in your bed. But there's one group of muscles that are not affected by this chemical, and those are your eye muscles. So your eyes are moving around because in your dreams you're looking around. You're walking around and you're looking around, and that's why your eyes are moving, we think. If you sleepwalk, you either don't release that chemical or it's not as effective. I don't know exactly. But you can just get up during REM sleep. If you're going to sleepwalk, it's most likely going to be during REM sleep. And you can just get out of bed and go do stuff. I mean, there's people who have driven places, cooked meals. I think that's just an excuse because I've woken up in the middle of the night and been like, man, I could go for some ramen right now or something. So I don't know if it's really true, but supposedly you can walk around and cook and clean and go on drives while you're in REM sleep. How crazy is that? Um, so anyway, if that's you or you have any family, comment, let us know. So that's it for sleep. It's always bad to end a lecture on sleep because it makes you sleepy. So we've got a few more slides. We're almost done. Language, we've talked a good amount about language. So if you're right-handed, in your left hemisphere, you have a Broca's area and a Wernicke's area. There's an R in here, I forgot. That's if you're right-handed. And these parts of the brain are highly integrated with areas of the brain that hold concepts and ideas. And if you think about it, that is one of the things that just makes us, and by us I mean humanity, so, I don't want to pat ourselves on the back, but great, right? Because we can have these ideas, and instead of them just being trapped in our mind, we can communicate them with other people. And they can think about it from a different perspective, and they can get back to us and say, here's why that's a bad idea or a good idea, or here's what you should tweak. And we can hash that stuff out. So that ability, that the fact that we can actually see the integration, this is not a hypothesis. We can do tractography with an MRI and see that the areas of the brain that hold concepts and ideas like the prefrontal cortex are highly integrated with Broca's and Wernicke's area. So that's pretty cool. If you're right-handed in your right hemisphere in the same spot, those centers are responsible for tone of voice and body language. And those particular sections of the brain are highly integrated with the limbic system and the cingulate gyrus in particular. So that when you're saying, hey, how's it going? You can say it nice, like, hey, how's it going, man? Versus, hey, how's it going? Like, how's it going? I don't know. But you get the idea. If you've ever texted somebody, you know that tone of voice and body language are important, because it's very easy. I've done this at work, like, hey, can you get that thing to me we talked about yesterday? Well, if you read it like that, like, hey, can you get that thing I've already asked you about yesterday or whatever, it sounds mean. But if I was saying it in person, it would sound different. And I don't want you to think I'm a bad boss. I'm a good boss. I don't know. I don't know if that's true. Ask somebody who works for me. Memory. We're coming to the home stretch here. Memory. Memory, to define it very simply, is the storage and retrieval of information. So we're going to talk about these things here. Stages, categories, and structures. So stages. The first stage of memory, assuming it's actually selected for memory, is called working memory or short-term memory. This is limited. This is not a scientific term, but it's the term that's used, limited to 7 to 8 chunks of information. And there's no better way to define it. It's so fascinating, because try to memorize an 8-digit number without chunking it up. And if you don't understand what I mean, think about a phone number. It's area code, first 3, last 4. That's how you read and remember a phone number. That's only 3 chunks, but it's 9 digits, right? So chunks is the only way it can be explained, because you can make one of those chunks bigger. You can maybe somehow figure out how to chunk 10 digits into one chunk, and then 4 digits into another chunk. And it has limits, of course. These people who memorize pi, it's like, man, how does that work? But anyway, we're limited to chunks of information. There's no way to other than chunks to describe it, because the same person can memorize, if you give them digits, if you just give them a paragraph of digits and say memorize this, they're going to count the first 10 and try to repeat it to you. But then if you say, no, take some time, group these 4 together, they might get much farther. So it's not a scientific term, but it's what it is. Long-term memory, on the other hand, doesn't seem to have a limit, but it does decline with age. So you do forget things as you age, and it's more difficult to convert short-term to long-term memory as you age. Sorry, we just talked about sleep, so I had a yawn. 5% of incoming sensory information is selected for short-term memory. So you remember about 5% of the things you see and hear, unless you're focusing on it. Then you're really going to hone in on that at the exclusion of other things around you. If you're just trying really hard to listen to my voice, you're not converting other sensory information to short-term memory, because you're focusing it. So what determines what is transferred from short-term to long-term memory? This is something you want to know if you're getting ready to take a test or study for boards or some license. You want to figure out, how can I remember this stuff? So it depends on a few things. Number one, and perhaps most importantly, is emotional state. The problem is it's tough to get emotional about anatomy or physiology or whatever. But if something is emotionally important to you, and it doesn't matter what the emotion is, it could just be fear of failure, Maybe that doesn't work as well as some others. I would think passion or something, really being excited about something would help you remember it. If you just think about, you drive by a car accident on the way home, you're not probably going to remember. If I gave you a quiz, you didn't know that it was a pop quiz. So you drove by a car accident on the way home, and I gave you a pop quiz, and I said, hey, what color was the car? Were the police there yet? How many people did you see in the car? Describe the damage of the car. Most likely, unless for some reason you paid really close attention, you even if you looked right at it, you looked right at it, you drove right by it, you probably wouldn't be able to answer those questions. But if for a moment you thought it might be the car of someone you knew, for example, a friend or whatever, because it was the same color and same model, and then fortunately it wasn't, you drive by and it was someone you don't know, and hopefully everyone's okay or whatever. But even just being emotionally invested for a few moments, if I gave you that same quiz when you got home, you'd probably say, well, yeah, so it was this kind of car and it was this color, and there was one guy in it and the police were already there talking to him. And so that might be a bad analogy, but the point is that emotion and memory are intertwined at a fundamental level. Of course, rehearsal has an effect on memory retention. That's the strategy we employ as students, is we try to rehearse, we study, we read again, we take notes. That's rehearsal. That's a substitute for the other ones. If you can't make it emotionally important, you rehearse. Association is a very powerful one, and this is one you have to try to employ if you can. It's easier to memorize something if you're associating it with something you already have memorized. You can anchor bits of information into your brain. The lab I'm getting ready to record is on the brachial plexus. I actually have you guys draw it first, because it's easy to just take that visual memory. You draw it 100 times, label it 100 times, get that down pat, because then when you go to a cadaver, which I will try to take you guys into the lab to see the brachial plexus at least, although it's looking like things are headed back towards lockdown potentially. But anyway, so you say you have this diagram memorized. It's a visual memory to you. Then you go into lab, and it becomes a lot easier to learn it on a cadaver, because you start to recognize patterns. You start to recognize things that look familiar. And now that new information gets converted to long-term memory like that. It's so fascinating. Of course, you have to take the time to learn two things in that method, but hopefully there's some things you already know, or even if you're learning things in real time, but you remember the one thing, then it becomes easier to go, oh, well, that new thing is right next to that other thing I just learned, so I just remember that. I associate them. So association is really powerful. And then the last factor is automatic memory, and this is just the idea that we can't figure out exactly why, but some things just automatically get transferred. And it could be some combination of the above three, but it just does seem like some things stick in your head without any obvious explanation. There are different categories of memory. Your brain treats factual memory, a declarative memory, different than the way that it treats knowledge, so conceptual understanding, and skills, motor skills. So you have declarative memories and non-declarative memories. A declarative memory is a fact. A non-declarative memory can be a skill. It can be a motor memory or an emotional memory. Something that's kind of interesting about the way your brain treats these is it stores them in different places, number one. But number two, one is contextually based. So declarative memory is contextually based. Non-declarative memory is not. So I don't know if you guys are super, introspective or not. If you're constantly analyzing yourself, hopefully you're not, because it's no fun. But I'm definitely in that camp. And so declarative memory is contextually based. What that means is it's very difficult to ignore the context in which you learned that thing or you memorized that thing. So for me, and then this is where the idea of like photographic memory comes from, because some people have a tendency to really heavily focus on the visual context of where they learn things. And I definitely have a bit of that. And I don't say that in like in a bragging way, because I don't feel that I'm any better a student than any of my classmates. It's just that's how I remember things. So if I read a book or take notes, when I take the test, I'm visualizing what I read or the notes. And it's not like I just have this photo and I'm reading the book again. It's just when I'm thinking about the things I've learned, I'm thinking about them in that context. You may be the same way, or you may focus on the auditory context. You may really remember things that I said really well versus something you read. So your brain might just be a little tweaked. Even when I'm sitting in a lecture hall learning, when I take the test, I'm still kind of visualizing the professor talking. That's because I'm just visually driven for some reason. But you might be auditorily driven, or you might be kinesthetically driven. And if that's the case, being kinesthetically driven in a lecture environment is difficult, right? Because you're not using your hands. There's no tactile feedback. In lab, you do great because you're touching the organs and you're touching the nerves, and it just kind of helps you remember it. So the only substitute, and it's actually a pretty good substitute, I employ it a lot because I'm visual and kinesthetic. I have no auditory retention of information. I could listen to a book on tape. I could listen to a professor. It's no good for me. But if I write it down or draw it, it's locked in. So figure out what you are. Figure out what works for you, and utilize that method. Non-declarative memory is not contextually based. So if you learn how to tie your shoes, you learn how to tie your shoes. You don't visualize the first time you learned how to tie your shoes. It's a motor memory in a sense. Although it's technically motor memory is a slightly different thing. A motor memory would be clearly there's a different, you've all experienced motor memory. You practice a sport, you practice riding a bike, your pathways from your brain that innervate your muscles develop and change. And part of that is based on memory. And you store memory near your primary motor cortex, motor memory. And then, of course, emotional memory, you can remember how things felt, how something made you feel. That's an emotional memory. Memory is stored based on regional need. So if it's a memory about how to perform a math equation, that's going to be stored most likely somewhere in your prefrontal cortex. All working memory actually is stored in your prefrontal cortex. But anyway, so it's stored based on where you're going to utilize it. The molecular basis of memory, difficult to study. We don't understand it super well. I mean, we understand the process and how memory is formed, like on a chemical and physiological level, but it just doesn't feel like we really understand it, right? I don't know. I guess that's a little woo-woo, but anyway, basically, when a memory is formed, neuronal RNA is altered. So new messenger RNA is delivered to axons and dendrites. Dendritic spines change shape. Extracellular proteins are deposited at synapses involved in long-term memory. The number and size of presynaptic axon terminals may increase, and they may release more neurotransmitter. What's interesting about this is this just sounds like a pathway strengthening. And maybe that's all memory is, I don't know. But this really just seems like a neural pathway is becoming more dominant, but maybe it's the same thing. That is it for this chapter. It's tough to talk about the brain without getting a little, philosophical there. I apologize. It's very interesting stuff. I encourage you to, if you decide to become, a professor or a researcher someday, the nervous system is fascinating. I am knee deep in fMRI studies right now. I'm working on a paper, and it's interesting. But you will start to feel like the more you learn, the less you know about the brain. It's one of those subjects. So I'm finding myself wanting to lean towards a different aspect, something that's a little bit more black and white. But anyway, for you guys, just more or less getting started on your journey, I wouldn't stress too much about this chapter. I keep it very conceptual. I'm getting ready to record a lab that will be a little bit more difficult, so I would encourage you to focus more on that this week. And Anyway, I need you guys to get your quizzes in by Monday, which is tomorrow. I'm recording this on Sunday, so let me know your final group submission for the quiz. I was reading the submissions and it looks like they were mostly right answers, maybe 1 or 2 on some of those initial submissions you might want to revisit. So get that into me Monday so I can give you guys all 100%, and enjoy the last few hours of your weekend. I will talk to you guys in the brachial plexus video next.